Okay, so I'm just going to very briefly introduce who we are and what we do, and then I'm going to hand over to Tracey to do all the work. Okay, so Tracey and I are part of the Colorectal Cancer Research Group at St John of God Subiaco Hospital here in Perth. This is who we are, and these are our main areas of interest. So most of our work focuses on identifying immune-related biomarkers of prognosis or treatment response in colorectal cancer. So quantitating cells in tissue is central to what we do. Back in 2017, yes it was, we were lucky enough to get funding to get some image analysis software. So at the time, we looked at the systems that were available, and we settled on StrataQuest. And I think initially we were drawn to StrataQuest. Tracey and I both have a flow cytometry background, so it was initially the flow style gating and back gating that Rupert just presented that really drew us to the software. And we also loved how customizable it appeared to be, and how strong the customer support seemed to be that they offered. So we've used StrataQuest for all of our image analysis since then. And Tracey's going to talk about one of our most recent projects that we've used StrataQuest for, and just share some of the tips and tricks that we've learned along the way.
Thanks, Mel. Hopefully I'll share some tips. As Mel just alluded to, so my previous life was as a flow cytometrist. I'm not sure I can quite call myself a flow cytometrist anymore, given that I haven't touched a flow cytometer in about 4 years, because now I play with images. So my role is also, I should also say, I'm not an immunologist either. So I guess I can just say I'm a cytometry nerd now. I worked 6.5 years in a core facility at the Centre for Microscopy, Characterization Analysis at UWA, where I did a lot of flow cytometry and microscopy. And when I came across to the research group with St John's, it seemed to kind of fit quite well to move into the imaging cytometry space as well. I also should note that when I first saw the scattergrams that Rupert presented, I was a bit sceptical as a flow cytometrist, but they're incredibly powerful and very useful, and they work very well. So just to give you a bit of context and background, the project I am going to actually talk about today is actually the PhD project of this very smiley, happy guy here called Ryan, who's not quite so smiley and happy because he's currently doing surgical training program, so he's not quite with us at the moment, otherwise he would be here today. His project is focusing on the role of the tumour microenvironment in metastasis development. So can we see a difference in tumours at the time of resection that might indicate whether they have a higher risk of going on to have recurrence? So to do this, and you'll hear lots of talks this week and last week at the cytometry meeting where we're now doing so many more colours. This is a relatively simple project. We don't have access to a spectral scanning system here in Perth, so we designed a panel to be run on a standard immunofluorescent staining 3D histec system, but we did get it working on the Bond Auto Stainer, much to Ryan's happiness, because it then meant that it was a overnight process instead of 2 days. And StrataQuest was kind of where I came in as well, helping to get some actual meaningful data. So very quick background, colorectal cancer that we work on, as Mel said, is one of the most common cancers in Australia, and it's actually one of also the highest mortality rates. So we often don't find colorectal cancer until it's too late. It develops, so colorectal cancer, we're focusing on the colon and the rectum, and it has different stages of invasion as it develops. So it starts in the mucosa, and we have different stages, so T1, T2, as it invades down into the bowel wall. And then eventually, once it leaves through the bowel wall, it will then move to the regional lymph nodes, and then often then to distant sites. In colorectal cancer, those are normally the liver and the lungs are the most highly likely. The reason I have the stage at the bottom here, stage 1 and 2, the stage depends on the level of invasion and where the cancer has gone. For the purposes of Ryan's project, he's classified stage 1 to 3 as the no metastasis group, and stage 4 is the distant Mets group. So stage 4 have Mets in another organ. For bowel cancer treatment, it depends often on the location and the severity of the cancer, whether or not they will have treatment up front. Many colon cancer patients will go straight into surgery. And then depending on what we find with the pathology and whether they're metastatic is whether or not they have adjuvant therapy, so post-surgery treatment. Then it's the waiting game for many patients as to if we do kind of clear all their tumour at the surgery, are they going to have a recurrence? So 1 in 3, unfortunately, will develop a recurrence, and that can be a local recurrence or that can be a distant metastasis. So the term that they've used here is metachronous metastasis. So when a metastasis has happened down the track within 5 years often, and it's happened, metachronous meaning it's happened at a later time point. So for some patients, we have a good idea that they're at higher risk of metastasis. So 60% of patients who do develop a recurrence, we kind of have a good idea from their clinical information and the pathology that they might have a recurrence. But it's around 40% of patients that we don't know that they're at higher risk. And those are patients that either we know to watch them more closely, or B, we can maybe give them adjuvant therapy if they weren't already indicated for it. So Ryan's project is focusing on the tumour microenvironment, which the immunologists in the room here will be quite aware of. And in the interaction, maybe there's a different immune profile of what's happening in these tumours. So Ryan's project is focusing on a couple of different panels, both focusing on lymphocytes. Specifically, the one I'm talking about today has a tissue-resident marker in it. So Ryan's aim is to evaluate the local immune response in that resected tumour and see if there is an association or a difference in that phenotype. So Ryan's study design is a nested case control study with patients that had surgery between 2010 and 2020. The main group we're interested in are these patients who have a metachronous metastasis. So at the time of surgery, they have either local or locally advanced tumours. They don't have anything in the liver or the lungs. But within 5 years, they then develop a metastatic recurrence. So these are the people that we really want to find something that might help us identify them. As a control group, the patients who don't have metastasis the same at the surgery, but also then don't develop any recurrence, these group of patients have a similar characteristic. So Ryan's identified for each of the patients in the metachronous metastasis group, another patient that has a similar age, sex, stage, treatment. And the third group is the synchronous metastasis. So these are patients who do have a metastasis at the time of surgery. So we know that they've already kind of developed into this stage 4. So these are our positive control, if you like. So that's the background, and this is kind of where I come in. So again, this meeting will see a lot of the amazing data. Today it doesn't have any data, I'm afraid. It's more just all about how we do what we do. The samples we're using are human FFPE tissues that we pull out from the pathology archives. We create tissue microarrays. We do our staining, imaging, quantitation. So I'm just going to talk you through a couple bit more details of those steps. The samples we're using, as I said, are these. We get the tissue blocks. Often there'll be multiple blocks from the tumour resection. Our pathologist will go through and she will painstakingly mark different areas. So we're interested in the leading edge of the tumour, the central tumour, and also the normal mucosa. And so she'll mark where these are, and then we'll try and line that up and punch a hole through and put that into a TMA block, sometimes more successfully than others. So we end up with a TMA that looks like this. This is actually one of our neater looking TMAs. Is anyone who's worked with TMAs, we all see these perfect little grids, and they don't always work like that. Sometimes you have like whole columns that decide to go on a little journey in a different direction. And also you'll notice for some of them, it doesn't look like we have kind of hit the tumour where we're supposed to. Some of them may be responders to treatment, and others we haven't quite got the right spot. So what we do is for each patient, we take 2 normal cores, 2 invasive margin cores, and 2 central tumours, because A, we miss the tumour, as I said, and B, the staining that we do is quite tough on the tissue, and we do lose cores sometimes. So here's just another image. So on the left, you can see those 2 normal cores, and the centre is the invasive margin, and on the right is that central tumour. So we also take serial sections for this project, so we have a H&E stain for all of our fluorescence, so we can actually look at that when we're doing annotations. This staining workup was on the Bond RX system, so I can say Ryan was quite happy he got this working. The system actually enabled us to get more reproducibility as well. We had a little bit less variation, but you might notice that previous TMA was a bit smaller than TMAs can sometimes be. So what we found is we could get an edge effect with these kind of systems where you have the solution flowing through. So we kept the TMAs a bit smaller and asymmetric to help us identify and orient them, and B, to be able to kind of reduce that. The staining method we're using is one that a lot of you might be familiar with. It's the TSA system, so tumorized signal amplification, where you have your primary antibody will go on, you'll have a secondary HRP, and then you'll come in with the floor attached. And that's quite a good system because you get signal amplification, so you actually get an increase in the signal. And then the good thing with that is that you can strip that off with another retrieval step, and then you can go in with another primary that's raised in the same species. So that was the reason we initially started looking at TSA, was so we could run antibodies from 2 or 3 mouse antibodies, but we actually have quite a good added benefit of having that amplification, which for our tissue, being colorectal, being high in collagen, we have quite high autofluorescence in the background, and that TSA signal amplification kind of helps. So as I said, we strip off the antibody afterwards, and then we go back in with another round. And so the panel that we have ended up with, we were hoping initially to get 6 colours out the system we were using, but really to get good accurate data, we ended up just settling on the 5, which seems very low compared to what some people are doing now. But we would prefer to get good data that was working well. So a little bit about the optimization. I'm not going to dwell too much on the optimization. It's something that lots of us now do, but it is really important to do it properly. Ryan actually wrote up all his methods because I think he spent about 80% of his time doing the optimization, maybe 20% actually doing the staining. So if you are interested in more details of what our process was, he's got that paper there. But basically it was the testing the antigen retrieval methods, the primary antibody titration, checking the secondary detection reagents. Some of them have got too much amplification, others not enough. Can we strip the antibodies off, and have we assigned the correct fluorophores? And the staining order is also very important. So I should note, we are using pathology samples. We don't have any control over how long that tissue sits in the formalin for. So if you're doing your mouse experiments, you can cull your mice, you have your protocol, 30 minutes, 1 hour, 2 hours. We are dealing with surgical specimens that they go into a pot in the theatre, they come down to the pathology lab, they might get processed the same day, it might be 10 hours later. So we have seen some differences in that fixation affecting our fluorescence intensity levels, and some particular couple of the markers. As I mentioned before, the tissue integrity, you're going through these rounds of stripping, you're basically heating you're boiling your tissue to 100 degrees. So we found that the slide choice was pretty important for us. We've used a slide, the Trajan Adhesive 3, I believe, that was super sticky, so they really kind of help hold on to that tissue. We also had an issue with antibody stripping, so I'll mention that in a second. I'm not sure, maybe we can turn the lights down a tiny bit for some of the images, I'm not sure. So we actually did our antibody titrations initially on DAB. Previously, we'd done it with fluorescence as well. We actually got a very easy switch over from doing the DAB straight to doing the fluorescence. And we did try where possible, actually for the most part, we use colorectal tissue. We have found previously that certain markers we would start with the tonsils as a workup because it was easy to get hold of. But we did find different staining intensity in the colorectal tissue compared to the tonsil, I think especially previously from PD1. So we then created these control TMAs that we could do this workup on. I mentioned the stripping, so we tested the 2 different stripping buffers that come with the bond, and so the stripping method essentially is a retrieval round, so you're putting your either EDTA pH9 or citrate buffer back on again, heating it up, and you're trying to get rid of it. I'm not sure how well this will show. Pretty much everything's stripped off fantastically, apart from, you can't see it very well, CD103. CD103 didn't seem to want to shift very much. So this was one that we went, well, we could sit there and try different methods, or we could look at our staining order and maybe pop it at the end so it's not really causing any problems if it hasn't stripped or not. I would say with this method, we also managed to compare also using a HRP inhibition step, a quenching step, which enabled us to kind of give the tissue a little bit of a break. And so we did that between, I think, a mouse and a rabbit round. The other staining order consideration is that this marker here, the Polaris 780, has to go at the end. So because CD103 had to go at the end and Polaris had to go at the end, they kind of had to go on the same couple. I'm not going to talk too much about the imaging other than just to show you the pretty pictures, but our imaging was done on a standard fluorescent scanner. So this has bandpass filters, LED light engine, and we designed as best we could around that. As I say, we could probably get up to 7 colours if we have a spectral, but we were pretty happy without getting any crosstalk with the markers that we have. And so we end up with this pretty thing that unfortunately is cancer. So people who come in often will see my screens go, wow, that looks really cool. Yeah, it's cancer. So very pretty, but we don't want it. But this is what we ended up with. So we've got our tumour infiltrating lymphocytes here, and we've also got some of the stromal T cells. So this is the part where we actually need to get some numbers out of this. So for us, we are interested in both the density of our different cell populations and where they are being presented, what kind of part of the tissue they're in. So Rupert's just showing you some screenshots of the very nice new version of StrataQuest. We're still using a slightly older version. So our original version that this analysis was done in was the 129. And the reason I mention that is I'll kind of do a side quest showing you a difference in the nuclear segmentation with the new version that we now have and that I'm now using on some phenocycler data. So our version's a little bit older. One other thing to note is what I'm doing today, it looks quite complex, and what I'm doing is actually designing my own apps. But when we first got this software, we basically gave these guys our stains and said, these are our markers, these are our questions, this is what we want to do. And they designed the profile for us with this easy user interface that we could then directly do the analysis ourselves. And so this is kind of the opening screen here with the apps and stuff that they designed. But now I am that nerd that pulls things apart, rebuilds them, and I've taught myself to make my own apps and profiles. So this is something I can do. And the nice thing that I can do is also make it easy to use for our other researchers that collaborate with us. I'll show you that in a sec. Rupert would have spoken about this. One of the things that actually drew us to the software was it will open many different file types and even the newer file types. So recently we've been able to use the PhenoCycler Fusion, develop 16-bit images, try to open it, didn't work, email these guys, oh, hang on a sec, we haven't had that yet. Well, 2 days later we get a fix, we get an update, and we can now open those files and analyse them. So I said before it can be quite complex. So here is what we call the advanced mode of our software, where I have all of these layers and all of these different engines and things that it's doing. But if I'm training up on a student to maybe check their analysis and maybe look at annotations, I can build this easy user interface so they can just click a few buttons and check a few things, and it will work out of the box, which is quite a nice thing to have. So my process usually is to open up the file that I have, to segment the cores, to classify the tissue mask. So if anyone is old enough to remember the old school way of doing annotation, sitting there with a little stylus and a pad and drawing around tissue to get areas, I don't have to do that anymore, which is fantastic. We are interested in classifying that tumour versus stroma areas. Then we need to segment our cells and determine what types of cells they are. So that segmentation process is highly important. The tissue microarray segmentation is very easy. It has an auto-detect, so if you kind of just want it to kind of find the tissue for you, it can. We tend to kind of create still a grid because we have an Excel spreadsheet with our TMA kind of map in it, and if we have every core that could possibly be on there, then when we export it, we can just overlay those really easily. So you can see also our TMA is asymmetric, which just helps us orient it depending on how it gets cut onto the slide. And also once these spots are created, you can change the size of individual regions and you can move them around. So usually I'll kind of put a rough grid on and then I'll just spend a couple of minutes just adjusting, going through and checking everything, and on we go. And what these regions mean is that you can apply an analysis for the whole slide, but it will export each of the data for each of those regions so that you only get the data for that kind of core, which is really neat, so we don't have to try and work that out down the track. Okay, so I mentioned the masks. The 3 masks that we tend to create are the tissue masks, so that we kind of limit our analysis just to the masks, the epithelium, which can either be the tumour or the normal tissue, normal epithelium in our normal cores, and the stroma. With our aim is to measure the area, and that enables us to give us the cell densities, and also then enables us to stratify the cells, whether or not they're in the epithelium or the stroma. So here's our little pretty core here. It's got all of our stains on it. The tissue mask that's overlaid here in purple, I'm not sure where the pointer is, I probably should be using that, is basically an auto-detect, and you can see there's a couple of bits that are cut out here. So even though we can do auto-detect, I'm still a little bit old school with this, we will still go through and check and annotate. So this one probably has a more annotation than we would need, but you can see here there was kind of a fold in the tissue, and unfortunately that's part of this. We want to get rid of those, because if you have folded tissue you're going to get erroneous results. Also, these are bowel cancer samples, and so we have debris, aka feces, in some of these, so those kind of luminal areas we want to get rid of as well, and so they don't kind of confound the results. So that will be our annotations. The next thing we want is our epithelial mask. Now, there is a classifier in the software. It's pretty good at actually pattern recognition now, as a pathologist I would do to kind of determine the tumour versus stroma. But because we've already had this process working and we have these profiles already existing, I've kind of just stuck with what we've been doing, which is to use cytokeratin, so it's a pan-cytokeratin marker to identify epithelium. We then do a threshold and detect on that, clean up that mask, and we create this epithelium mask. So here in turquoise is our tumour, and anything that is green is in the stroma. And as I said, we can measure the areas of those. And then Rupert briefly showed us the distance masks as well. We can choose to create these distance bands and look for the cells that exist within those bands and distances from the epithelium. And this is the reason that we do this as well, is that in yellow here are all the nuclei that are on that epithelial mask, and in blue all the nuclei that aren't. So we can look at the number of cells in the tumour versus the number of cells that are in the stroma, and also we could do that in those distance bands as well. So the bit that is really important that lots of people who do this type of work will tell you is that, how do you actually determine what a cell is? And there are always caveats with this. We are looking at 2D sections, we are looking at TMAs, we're not going to get this absolutely perfect. In fact, that's been a lesson in non-perfectionism for someone who's coming from flow cytometry. You have to get a lot less perfect, but there's lots of things we can do to reduce that error. So here's our all stain sample, and this is our DAPI channel. Again, can we put the lights down slightly, because there's a few fluorescent images coming up. So this DAPI isn't particularly pretty. We actually had, again, we see a little bit of sci-fi kind of spillover into here, a little bit of autofluorescence, but we can actually do some corrections and remove some of that background and remove some of that kind of it's almost a compensation, crosstalk. We also found that this is showing us the original segmentation that we would do for the nuclei. Bianca showed us that if we create some filters on the DAPI staining, we can then get an improvement in our nuclear segmentation. I will point out our samples are pretty tricky. We're looking at colorectal tumours where we have a really big range of nuclei shapes and sizes. So the tumours can often have these really elongated, almost kind of oblong nuclei. The lymphocytes, we know, will often be nice and small and round, unless they're in the stroma, when they can be a little bit more elongated and stretched. And then you can also have high dense areas of lymphocytes as well, which can be tricky. So we're never going to get our nuclear segmentation with this method completely perfect, and we chose to focus more on settings that would better identify our lymphocytes rather than the tumour nuclei, because we're not so interested in actually the tumour itself. And we got it to the point where we're pretty happy with it. I'm going to do a very quick side quest, because we have now got a new version and a new algorithm that has greatly improved, I think, that nuclear segmentation. I know Rupert's team worked quite hard on this. So this is a similar area. I did this very quickly on Sunday. I kind of went in going, I should show you guys the DNN for this one. So the deep nuclear learning, this is our original nuclear segmentation on this sample. So you can see we have a bit of over-segmentation, and it's not perfect. It's good, but not perfect. Now with the DNN, we get a tighter, rounder nuclear segmentation, and you can see we've got more accurate identification of the individual cells over here. There's still a slight bit of over-segmentation, but that's much improved compared to what we had before. So I've just got a couple of overlays just to show the difference between them. And so you can see also that the new DNN is a much tighter nuclear mask, which is kind of important for the next part of the analysis. So for each of these circles here, that's classed as a cell event, and the software will spit out whatever data you choose to look at. So you can choose to look at any of the other markers or colours or masks within that nuclear envelope, but we're looking at cell surface markers, so we don't really want to be looking inside that nuclear envelope. I've also got, this is just showing that on the left, the DNN on that whole tissue as well. So you can see it in the tumour versus the stroma. Okay, so I'm going to go back to my original nuclear segmentation, which is what we did this analysis with, because I haven't quite got the next project ready. So how do we look for the cell surface membrane? Well, we create a cell mask. So this is still the DAPI channel, but here we've actually grown that nuclear membrane, that nuclear mask outwards and inwards a little bit. And so we end up with a mask. This is now where we can look for our actual marker of interest. And so, and to know kind of where do we put this, because again, I have all the flexibility deciding how much do I grow it out and how much do I grow it in, is I will usually use a marker I'm looking at. So I look at the actual, this is CD3, and I will then say, right, is my CD3 mostly sitting within where that cell mask is? Am I happy with it? And then we go on to the phenotyping. So for each of those cell membrane masks, you can then look at the fluorescence intensity in them. And there's a whole range of data that you can output. You can right-click on any one of these cell masks and say, show all the data, and it will show you every bit of information. You can look at mean intensity. You can look at the 75% upper mean, like the brightest 25% pixels or the brightest 50% if you want to ignore if it's a very big area. And you can look at the size as well. You can look at the fluorescence intensity, just kind of look at the brightness. So the old school kind of method that we started using, which is to look at the mean intensity. So here's the CD3 mean versus the CD8 mean. And you create a gate here, and then you can backgate and see where they all are. I haven't got a screenshot here, but one of the really nice things as well is that you can actually do a validation. So it will bring up another window, and every one of these cells on its own, you'll have a little image of, so you can kind of scan through and go, am I in the right ballpark? So to set my cutoff, I'll generally create a little kind of gate at that threshold, and then I'll say, am I getting my positive cells or my negative cells? And again, you can set that for the whole slide, and on you go. So for the double staining, because you have CD3 and CD8 here, you can then gate around this top right-hand side. Apologies, the colour change. And now we're looking at the double positive CD3 and CD8s in here. So I haven't necessarily picked the best example of this, but sometimes we are looking at rarer subsets, and I am a skeptic, and I want to be very confident that when we're looking at a double positive or triple positive cell, that I'm really looking at double, triple positive and not just a neighbour that's nearby that happens to have had a bit of its cell membrane caught up in the mask. So this is that kind of standard method of doing the gating. And then I kind of thought, sorry, that's just both of them together. And then I thought, well, can I get it better? What am I normally doing if I'm kind of flicking between channels to see what is positive and what's negative? And I kind of think, well, I'm looking to see them overlapping. So if I create a mask, and this is something that I've just done myself, if I actually make a threshold mask and say anything that has a threshold of intensity higher than a certain amount will now become this mask, and wherever it's green, it's actually a value of 255 versus 0, so it's like a binary mask. I overlay those, and now I can see anywhere that's yellow, this is where I know I've got both CD3 and CD8 stain. And then I can look at how much of this actual mask is within my cell membrane mask. And I can get some improvement. So here I've got the sums, so the area of that CD3 and CD8 mask. And now when I backgate, I can see that I'm getting a slight improvement, or I'm certainly getting, especially my triple stains, I'm more confident that I'm getting the double or the triple stained cells. So it's pretty subtle between these images to kind of see what the difference is. But when I'm using the mask, I'm kind of avoiding using the one that's kind of too close by, and I'm a little bit kind of more stringent with my gating. So just coming towards the end now. So to end with this project, we will end up with a total count of our CD3s, our CD3, CD8s, 3 103s, and the triple stain. We get the number of those different cell populations within that tissue mask compartment. We get the number within the epi mask, the stroma mask, and then we get the areas of those. But the other thing that we can do, because of the hierarchical gating with the scattergrams, is we can look at the proportion of those cells. So it's not just how many of the cells are in there, it's what proportion of the CD8 cells are expressing CD103. So our aim, and I said before, I'm sorry, I'm not going to show you any data. The aim was to determine if there was a distinct immune subset in the metacrinous metastasis group. We did this using this 5-colour panel with a couple of different panels. We did it using the StrataQuest, and we do have a little bit of preliminary data that we've got on a poster on Thursday. So if you wanted to come and see what we did actually find, then you can come along and have a look at that.